for a healthy and a sustainable diet. It's organized by two organizations, the Swedish Royal Academy of Sciences and then the National Committee on Nutrition and Food Science, as well as the National Research Institute, mainly financed by Formas, which is called Plant-Based Proteins for Health and Wellbeing, shortly PEN Sweden. So we are very happy to jointly host you for this symposium. My name is Robert Brummer. I'm director of the PEN Sweden and professor of gastroenterology and clinical nutrition at Örebro University.
And my name is Nicklas Neuman, associate professor of food studies, nutrition, dietetics, Uppsala University, and work package leader for both periods of PON.
Plant-based proteins play quite a pivotal role in the system change of our food system towards a more sustainable system, also healthy for the planet. But there are some issues associated with that, and there are exactly that kind of issues we will discuss today. What is the role of plant-based proteins in a more broader perspective for planetary health? What are the health challenges we are facing? But also what are the technology and also consumer preferences issues we are facing with the change to more plant-based protein consumption. And we start with more global orientation.
Yes. Without any further ado then, I would like to present our first speaker, Line Gårdang. Line is the Kurt Bergfors Professor in Sustainability Science at Stockholm Resilience Centre, Stockholm University. She also directs the Stockholm Resilience Centre. We have invited her because of her important contributions to interdisciplinary research on sustainable food systems, which the most important, or at least the most famous example, of course, is the EatLancet reports, or the two EatLancet reports, and these will be at the core of her talk today on global perspectives on plant-based proteins. So warm welcome to Line.
Thanks a lot for inviting me here today. And as you said in the introduction, I will talk about the EatLancet Commission. I assume that most people in the room are familiar with the EatLancet Commissions. The first one being published in 2019, and then we came up with an update just a couple of months ago, so the 3rd of October. And I was part of the leadership team leading the chapter on transformative change in the Commission. So what is a Commission? Well, first of all, the overall kind of tagline of this 2025 Commission was healthy food, accessible to all, produced, processed, distributed and consumed fairly within the planetary boundaries. So if you compare to the 2019 Commission, you can see that this 2025 Commission puts a much stronger emphasis on justice and fairness in terms of food system transformation. The Commission consisted of 24 commissioners representing different countries and many different disciplines. We gathered people from nutrition sciences, agriculture, sustainability sciences, economics, and sort of interdisciplinary skills. It was chaired by Johan Rockström, Walt Willett, and Shakuntala Tilstedt. In addition to this, we also had a very good and important young research group, about 10 people, postdocs and PhD students that supported the work of the Commission. We also had the possibility of inviting other expertise where we felt that we needed to sort of bridge gaps in our analysis. So all in all, 70 people worked on this commission, representing 34 countries and 6 continents. So it was really a global effort. The Commission had 5 different working groups, and I like to think about it as 5 groups and 3 key pillars. So the first one was on health. What is a healthy diet? The second one was on the environment. So what is an environmentally okay food system within the planetary boundaries? What is a just food system is the third pillar. And these three pillars were done independently of each other. So that means that when we looked at what is a healthy diet, we only looked at it from a human health perspective, not whether this diet was actually environmentally friendly. So, same thing for the environment. We looked at a food system, what is required of a food system to fit within the planetary boundaries if you only look at the environment. And then similar for justice, what is a just food system? And then we modeled whether this is possible. Is it possible to achieve this healthy diet within the planetary boundaries and in a just way? And then we looked at what type of changes are required in order to do so. So I'll go through these different sections to some, at least, extent that's possible within the next 25 minutes or so. So first, a healthy diet. For everyone who read the 2019 report, the healthy diet will be very similar. There's very little change to what we published in 2019. And that is not because we haven't engaged with the evidence that has come out, because that was really the main thing that we did over these past years, is to check what is the evidence that has come out between these two different reports. But basically, the evidence basically confirms that this is a good way of defining a healthy diet. So if you look at the overall plate, basically half of it is filled of fruits and vegetables. There is a big share of whole grains. So eat most of the grains as whole grains, basically. And then big rooms also for nuts, for legumes, but also small room for animal-sourced foods and some fats, especially from plant fats, unsaturated plant oils, and also a little bit of room for sugar. One can also think about this as different servings per week. So if you translate this in terms of what would it look like in your sort of diet for a week, basically it says eat vegetables and fruits 5 times per day, nuts around or seeds around 1 to 2 servings per day, and legumes, eat it every day. So that's, I guess, a big take-home message also for this crowd. In terms of animal sourced food, the diet says around 2 servings of poultry per week, about the size of a hamburger of red meat per week, or 2 eggs per week. It's hidden here, but it also says fish and shellfish somewhere 2 to 3 times per week. The diet is an omnivorous diet, so as you have seen in these previous slides, it can be eaten with both plants and animal-sourced foods, even though the animal-sourced foods are in limited amounts. But we also show that it's also possible to eat this either as a pescatarian diet, a vegetarian diet, or a vegan diet. So it does have the flexibility to also encompass these different types of dietary patterns. But of course, if you go towards a vegetarian or vegan diet, just doing that and limiting the amount of animal-sourced proteins doesn't take you to a healthy diet. This is really about finding the balance and finding the other type of food groups that you then need to increase. And if you go to vegan diet, you may also need some fortification in your diets. There's been an analysis of what would happen if people shifted from current diets to this planetary health diet. And what we could see is that basically it would be a reduction in premature mortality with 27%, or 15 million deaths per year could be averted on a global scale. sorry for the Swedish, I miss this. But I wanted also in this context and in terms of plant-based proteins and what we're talking about here, I also wanted to show the different types of food groups that you see here and what we are currently eating today. So it's a bit of a messy figure, but you see for all the different continents, how far away we are from the reference diet. The reference diet being this dotted line here, the 100 level. And I thought it was interesting that the group where it's, well, the two groups that it's really the biggest gap in terms of under-eating today, it's whole grains. So we eat enough grains, but we don't eat them as whole grains. And then it is legumes. So here's a big need to increase legume consumption in general. So that's what it says also in Swedish for the non-Swedish speakers. We do emphasize, I think the first EatLancet got a lot of criticism because it was perceived as a one diet fit all type of approach, that this is a way that we're emphasizing everyone should eat in the same way. That's definitely a misunderstanding of the planetary health diet. We're really emphasizing the importance of choice, of diversity, of adaptation to different cultural contexts. The ranges that we have in these different food groups also allow for that type of adaptation to happen. So in this year's report, it's much more of an emphasis of that diversity and adaptation. And I think it's interesting being in Sweden, that in Sweden we already have a process, I think, that is fairly good and very similar to the EatLancet that takes us to a very similar diet. And that is the Nordic nutrition recommendations. And in many ways, and sorry again for the Swedish, I couldn't find this in English, I'll go through it for English speakers. But in many ways, the EatLancet diet isn't so different from what the Nordic nutrition recommendations say. They also emphasize a primarily plant-based diet, lots of vegetables, fruits, berries, legumes, potatoes. Well, maybe we don't emphasize potatoes, but anyway, and whole grains. Rich in fish and nuts, limited amounts of red meat and chicken, moderation of dairy products, and minimal of processed meats and alcohol, fat, sugar and salt. And in Sweden, this has also been translated into Swedish dietary guidelines. I think some also misinterpret the EatLancet as trying to be a dietary guideline. We don't have any, we're not assuming that this is a dietary guideline. Dietary guidelines needs to be adapted by the people that are going to use it. So in Sweden, the National Food Agency is the one who are, based on the Nordic nutrition recommendations, developing the dietary guidelines. And it's also very similar in many ways, with a little bit of difference between some of the numbers. But the basic directions to where we need to go is similar to the EatLancet Commission. Here you also see in the Swedish dietary guidelines emphasis of beans, peas and lentils, that they are emphasizing eat that every day. So that was basically the diet part of the EatLancet Commission. I go in quickly to the food system shares of the planetary boundaries. I'm not sure how familiar you are with the planetary boundaries in general, but the planetary boundaries globally is an identification of 9 different variables that, if these are transgressed past a threshold level, it risks destabilizing the Earth system that is so important for human well-being. So here you have the different variables. It's climate change, land system change, biodiversity, ozone depletion, biogeochemical flows like nitrogen and phosphorus, freshwater, aerosol loading, and novel entities. Novel entities being things that haven't existed in the biosphere before, like pesticides or antimicrobials. the control variables you see here, and then what the EatLancet Commission did was to identify how big share of the overall boundaries should be allocated to the food system. Because of course the food system, we need to eat, we need to produce food, so some impact on the earth system food production will always have. So we identified the shares that we could allocate to the food system, and then we analyzed then how big share is it today and how far do we need to change. So this is what how much society today transgress planetary boundaries when you account for all human activities on the planet. So what you can see here is that the planetary boundary is the yellow circle in the middle here, and then it's a high-risk line being the orange, and then you can see for all of these variables how far we're actually outside of the planetary boundaries already. And this is accounting for everything: transport, energy use, industry, etc. We then looked at what is the share of food systems impact on these planetary boundaries today. And as you can see, the food system is a major driver for the transgression of 5 out of the 9 planetary boundaries and 5 out of the 7 planetary boundaries that are already transgressed. It's also a big contributor to climate change and to ocean acidification. But what this means to me is also that food is the core solution. If we're going to live a good life on this planet, within this Earth system, we need to find the solutions in the food system. Because even if we can take out fossil fuel, which we really need to, from other sectors, we will still have big transgression of all the other planetary boundaries. And we need to focus on the food system in order to do so. So this is a very global analysis, obviously, but it draws on local data. So we use different data sources from around the world, and I'm just giving a few examples of these data sources. But here you see, for example, for pesticide risk scores, and you can see how unevenly these are distributed globally also, with some regions having really high impact and others a bit of a less impact. And then here just another of these maps that are used with local data analyzing the impact on the planetary boundary. So here is for nitrogen surplus, so it's nitrogen leakage from agricultural land around the world. I will jump to the modeling section next, because in the 2025 analysis, we managed to gather 10 different global modeling groups around the world that all did the same type of modeling, where we assessed then, is it possible to produce this diet for everyone within the planetary boundaries? And that's a major effort just to bring such a big team together to do this type of modeling. But that also means that we couldn't do the modeling of all the different things that we would have liked to do and at the detail level that we would like to do. So I will in a little bit present also the justice part of the EatLancet Commission, but we were not able to do the modeling in the same way for the justice variables. But here, think about it as this kind of healthy diet within the planetary boundaries, basically. So first of all, in the modeling section, we looked at what we eat and how we can shift these diets, but also what and how we produce these agricultural goods. And then looked at food loss and waste, so what do we throw away? And then we also had another scenario that linked that to other climate mitigation efforts. So whether when we're shifting these things, are also phasing out fossil fuels, for example, in other sectors. So we combined these 4 different elements in different scenarios. Here you can see just in terms of if we were shifting to the EatLancet planetary health diet, what are the different food groups that we would need to increase production of or decrease production of. And you can also see the reason these bars are so big is because we had these 10 different modeling teams. So the models have a lot of uncertainty in them. So these represent kind of the span of the different results from the modeling teams, with the white lines being the average across these models. But I think for these groups especially, it's interesting to see that the one food group that we really need to increase production of is vegetables, legumes, fruits and nuts. It's a big one category when we combine the modeling results, because not all models can kind of distinguish between these categories. But you see the big increase in production that is needed globally to eat a healthy diet worldwide. Well, you see for other food groups like livestock, ruminant meat, for example, it is a reduction of current production. Also a bit of a reduction in sugar, but overall sort of a bit of an increase of overall production from agriculture, and especially from this vegetable legumes category. Here's another way, because we could break out from one of the models just to look at the legume production, for example, and I think that's quite interesting. What you see here is the white lines. You have sugar also. Those are the two graphs that I could find for this presentation. But you can see the white line is the trend line from 2010 till 2025. And then business as usual. Like if we do nothing, where are we going? You can see a slight increase in sugar production overall. And then legumes, you can also see that it is going up production-wise, but at a very slow rate. And then you can see the gap to, if we are to achieve the overall consumption or production that can meet a healthy diet, it's a big jump and increase in production that is required from legumes, but also a possibility of actually cutting sugar production. We then looked at what will be implications for the planetary boundaries. And as I showed you already, we're already transgressing most of the planetary boundaries. But if we would have just a business as usual development in the food system, we would see a further increase of land for agriculture, a further increase of greenhouse gas emissions from agricultural systems with 33%, increase in blue water, nitrogen use, phosphorus use. If we shift to what we call the EatLancet scenario, and this is a shift just to the diet, cutting food loss and waste by 50% and improving production practices, we could reduce the need for land overall globally, and we could cut greenhouse gas emissions with 20%. But we still see a little bit of extra need in blue water use, nitrogen and phosphorus. We also ran another scenario, as I talked about, where we see climate mitigation from other sectors. And if we combine that with the EatLancet scenario, just focusing on the food system, then we could cut these emissions further. So basically the sort of key takeaway from this part of the analysis is that we really need efforts in the food system and also outside of the food system in order to reach the needed benefits. These models, as I said, they're fairly coarse, especially if everyone is going to run exactly the same scenarios. We also did an analysis, a synthesis of different types of agricultural practices that could further improve the environmental benefits. And we looked especially at what we call ecological intensification practices. And these are things like crop and non-crop diversification of systems, organic amendments, reduced soil disturbances, and so on. And then we checked how much that they actually could improve different environmental variables. I'm not going to go through this, but basically it shows that doing more ecological type of intensification practices would help substantially and beyond what we could see in these scenarios that we modeled. So that was a bit on the healthy diet, the planetary boundaries, and whether we can do it within do this within the planetary boundaries. What the EatLancet Commission also did that was new this year was to add this dimension of justice. And this was an enormous effort where we really tried to sort of address it across multiple different dimensions. So we developed a new framework for analyzing justice of food systems, where we looked at 3 dimensions of justice: recognitional justice, which has to do with whose identities, values, cultures, and experiences are acknowledged in the system. We looked at distributional justice, which has to do with who gets what and how fairly are different resources, risks and benefits shared in a society. And then also representative justice, which has to do with who actually has a voice in decision-making processes. When it comes to the planetary boundaries, for example, or even the healthy diet, there is a lot of scientific evidence of where are biophysical sort of thresholds where we know the Earth will start behaving in a different way, or our bodies will react in a different way. In terms of justice, we have fewer frameworks to decide on what is actually a just system or not. What we did here was to lean into the human rights, as the human rights are something that are, at least for many countries, agreed upon. So we also looked at these different dimensions of justice and then used the human rights approach of right to decent work, right to food, and right to a healthy environment, as these are ones that are really related to the food system. We also emphasized freedom and agency. And that led us to defining 9 different variables that we could, for 6 of them, we could quantitatively assess whether the current food system is just or not. I'm not going to go into all the different dimensions here, but mention them here, and for the people who are more interested, I really recommend reading into the publication. But for right to food, we look at affordable, healthy diet. So whether this planetary health diet is actually affordable for people around the world. And what we could see on the data is that currently 2.8 billion people globally cannot afford a healthy diet. We then looked at what we call a healthy food environment. So that is, are we living in food environment which are conducive? Is that the right word? Like where it's easy to make a healthy choice versus an unhealthy choice. I often like to think about this study that came out from Karolinska Institute, and I think Hjärt-Lungfonden, where they gave cameras to kids in Sweden, and they had to take photos of all the advertisement they saw on food. And then they looked at the advertisement and said that 80% of all the advertisement is just junk food. So today we live in a food environment where it's not easy to make the healthy choice. And we doing that, we found that 5.6 billion people globally today live in food environments that are unhealthy, or are sort of supporting more an unhealthy diet. We looked at the right to healthy environments. That is, if you think about the planetary boundaries, they are at a very global scale, right? But how does that impact your experience in a local environment? So, for example, for non-toxic environments, it's if you're living in an area where you have experiencing a lot of nitrogen pollution that are impacting your local environment. And we looked at a safe climate being where people are suffering health extremes from extreme heat waves. And then the right to decent work, a living wage, and meaningful representation. So when you look at this, you can see that there are a lot of people around the world who are living below these sort of social foundations of a just food system. We also looked at how many people are eating sort of a diet that has a higher impact on the planetary boundaries than the fair share of a per capita diet. And when we combine this, that it's both the right to food and the right to this, to healthy environment, the right to decent work, but it's also a responsibility of eating a diet that is kind of within the planetary boundaries. We could see it's only 1% of people around the world that lives both sort of within social or above social foundation and below these planetary boundaries. We also identified these variables for justice as important for a just food system, but didn't have the capacity in this year's EatLancet Commission to actually quantify them. But one is freedom from corporate control, because we can see that when corporations get too high market power, they also often lobby for sort of more for maintaining profit than actually for health or sustainability. That doesn't mean that the food industry is not an important part of the solution, but market concentration can have a big impact of people's individual and collective agency. We also emphasized civil and political freedoms and non-discrimination. Yeah, I mentioned this, only 1% of the population living kind of within this safe and just space. And then just to sort of conclude, we also looked then at solutions and transformation. And I showed part of these graphs before, but just showing the big difference between where we are going right now in terms of business as usual and where we actually need to go for a few of these variables. So when we talk about the food system transformation, it's basically reducing this enormous gap between business as usual and where we need to be in the future. What we did is we developed something we call the transformation wheel of solutions and actions. So we started individually for first the diet. What is it that we need to do and how can we do that? And then we looked at so first for the diet, then for the production systems, and then for getting into a more just food system. And when we say what do we need to do, we call this the solutions, and then how do we do it? These are the actions that we are identified. And we try to look at we looked at the sort of summary of literature on where do we see evidence for what types of actions are required. I'm not going to go through all of this. It's basically 8 solutions and 23 actions. But in terms of dietary change, we talk both about the shift to these healthy diets in areas where we don't eat a healthy diet currently, and the need to protect and promote already existing healthy diets in areas where people are already eating a healthy diet. Because we see that there is a big trend in also abandoning already existing traditionally healthy diets. In terms of shifting to a healthy diet, we emphasize 3 different actions. The first is price-based interventions. So it's things like shifting taxes, reducing taxes on unhealthy food items and reducing taxes on healthy food, increasing on unhealthy food, so that you shift the relative affordability of the healthier food. We also emphasize regulating marketing and the potential of warning signals on unhealthy food items. And then there's a lot of focus on just making food cheaper for improving affordability of food, but what we emphasize is also importance of increasing purchasing power, especially of the lowest quintile of households through different types of social protection mechanisms. And we have similar actions suggested also for production systems and then also for the justice of food systems. I'm not going through them here because it's a bit overwhelming. What we also emphasize is that these actions should be thought of as coherent bundles of policy. So not just implement the tax on sugar, for example, but really do it together with reducing taxes on the unhealthy food items and together with restrictions on marketing. We also emphasize the need of building coalitions of diverse actors to come together to be able to achieve these different goals. But in order for these coalitions to be successful, they especially need to address 3 different key barriers, and that is insufficient political leadership, corporate interests that are blocking change, and weak and fragmented public demand for action. And then we also emphasize the need to unlock financial resources, because we know that this transformation will be costly, but other type of analysis have shown that basically 1 krona investment in the food system transformation will reap around 10 times those benefits economically because of the enormous cost that the current food system have today in terms of decreasing public health and a bad environment. So we see that there is already a lot of funding in the system, and this needs to be redistributed. So these are things like repurposing existing subsidies in agriculture. Instead of subsidizing the things that we don't need increased production of, subsidize the more healthier crops that we need to increase consumption of. So maybe move into the legumes and the plant-based proteins rather than animal-sourced proteins, for example. We emphasize the need to align with other policy priorities. If you look at global climate mitigation funding today, for example, it's only 4% of that funding that goes into the food system, despite the food system being at least 30% of the greenhouse gas emissions. So how can we basically raise the voice of the whole food system, because we also need to take more part of those investments. Now it's also the security is high on the political agenda. Transforming the food system for health, for sustainability and for justice will build a more secure society. So can we also get more of that funding into the food system transformation? Attract private funding as well is important, and then finally increasing international development efforts, because there are lots of countries where the need for investments is beyond what they can currently fund. And that's basically the end of the EatLancet Commission presentation. We do emphasize, and I'm sorry about these different little things here, they're in different places, but I think you can read anyway. Food systems cannot be just without ensuring that the planetary health diet is affordable and accessible to all, and without substantially reducing the transgression of the planetary boundaries. But justice is also necessary to overcome the barriers that currently impede transformative change. Justice is therefore not only an outcome of the transformation, but a prerequisite for enabling it. So that's it. Thank you.
Okay. I'm waiting on my own, but okay, I can start it now. Thank you very much for your presentation. I will ask first whether there are any questions in the hall. Otherwise, I can start with one. You gave the broad picture. If we go focus on the area of plant-based proteins, what kind of contribution does that have if we change from according to the indications you give,
Yeah.
would it make really a big change, or is it just a marginal change? Or?
I think that is would be the largest, in terms of at least environmental impact, that would be the largest change that we would see. I mean, I think from a health side, I think whole grains, for example, is also really important. But from the environment, that is the biggest leverage that we can see. And it is both because of the animal-based proteins having such a high impact on the environment. Legumes and legume production also have important components like improving soil quality and nitrogen fixation, etc. So it could also reduce the need for fertilizers, for example. So it really has a big leverage. Mm-hmm. Yeah. Mm.
Yeah.
It's a bit of a detailed question, but in one of the slides that you showed, I think it was on pesticide and nitrogen, for example, Sweden looked relatively good. Do you think, is that related to climate, that we don't need so much pesticides, or is it related to that we actually do not produce enough food to feed Sweden, so to say? Do you know what it is related to?
I mean, I think there are a few different things, and I haven't dug into that. I mean, sometimes with global data sets, it also gets a bit blurred. But I would say Sweden has a fairly strong or good restriction on pesticides, so that could impact that we are relatively low. We also, Sweden is a big country where most of the production is in the south, so that of course in the map it also looks quite good because we don't have so much production up north, and there are other areas where it's much more intense production as well. So I would say those are a few reasons, but we also do have fairly good production practices here.
I have a question.
It's a sort of assumed that a healthy diet is expensive. But I would like to question that, because I think in the Nordic countries a healthy diet is not more expensive than an unhealthy diet. But it could be, of course, different across the world.
Yeah, no, I completely agree. I think here it's not, and there has been others that have shown that. I think it is possible to eat a healthy diet, a plant-based healthy diet, at actually lower costs than what people are spending on food right now. So it is definitely, it doesn't have to be an expensive diet. If you look globally, it is a lot of people still that can't afford it. I think sometimes the EatLancet Commission is also, people are criticizing the development of a healthy diet that we are proposing, as it's not affordable then for everyone. On the other hand, I think this is what we need to try to achieve, and then we need to focus on how do we achieve that. And I think that has to do also a lot with this repurposing of funding, and how do we actually invest in what type of agricultural production. It's also about bringing up purchasing power rather than just making a healthy diet cheap. So with all these combined efforts, it's definitely within reach to make the healthy diet affordable for all.
There is a question.
Yeah. Thank you for a great presentation. I just had a question on the meat categories. Sometimes they're reported separately in the analysis, and sometimes they are put all together.
Yeah.
They have a different kind of impact on the environment. So I was just curious on how you reflected on that.
That's a big. That's an important point, and we try to keep them in the table. In the planetary health diet, we differentiate between red meat and sort of white meat and fish and seafood. And then in the analysis, in the modeling analysis, it depends a little bit on what the models could capture and how they could do that. So I think that's why the categories sort of change a little bit in the different throughout the publication. But it was something we tried to kind of differentiate, because they do have very different both impact, both health-wise and environmentally.
I saw in one of your slides, which is a sort of a common hypothesis, is that corporations or industry are hindering the transformation. I would say that they are necessary for a transformation, and a lot of organizations actually stimulate also the conversion to a more healthy food system. What's your opinion about that?
No, I completely agree that I think they're totally necessary. Without the industry, we can't shift this food system. So we really need to work together and join hands together. That doesn't mean that they're not lobbying against what needs to be done, or some other organizations are doing that. So I think that is why I think.
But that is more globally. I think in the Nordic countries it's rather the other way around.
I hope so. Yeah. I think it is. I mean, I think it is. How to say this, but I really do think we need to work very closely with the industry, and they have such a big role to play. On the other hand, it also does worry me if I look at, for example, the Climate Policy Council that is put together in order to also check what is the government's policies on climate actually achieving the targets that we have and leading to the outcomes that they're saying they have. And that is the Climate Policy Council is just academics sitting there. We have the same Finance Policy Council, and then we have the Food Policy Council, and that's only industry. There's no academics in there, and that does worry me. That it is. So where is the academic voice that also has an capacity to check, are we aligned with the overall targets and goals? And that doesn't mean to be anti-industry, but it's just we need to also have a balance of the voices.
And there are also, of course, a lot of public organizations who are stimulating this transforming, like schools, for example, and so on. They do a tremendous work on actually changing attitudes and so on.
Yeah, exactly.
Okay, thank you very much, Nina. You will get a present also before you leave. And then we go over to a more individual part, the health, individual health, the second session of this symposium. Now we have spoken more on globally and on level of nations and so on. Now we will go more to also the level of individual health. I would like to introduce Anna-Maria Pajari from Helsinki University. She's a professor in molecular nutrition, but not only working on the molecules, I would say. She's doing a lot of research on the dietary intervention studies for health, both nationally as well as also on a European level. So Anna-Maria, the floor is yours, and looking forward to your speech.
So good afternoon, and warm thank you for inviting me here. So I slightly changed my title to include benefits as well as challenges, because it's obvious that we have benefits related to plant-based diets and higher consumption of plant-based proteins. Today I'm briefly first describing the nutritional contents of plant-based proteins, and then mostly I concentrate on showing you results from 2 randomized controlled trials where we have replaced plant-based proteins where we have replaced animal-based proteins with plant-based proteins. So the trials are called Skenoprot and Beanman. The first one is about replacing. So the first one is about replacing red meat, poultry and dairy products dose-dependently with a variety of plant protein sources, and the second one concentrates on replacing red and processed meat with legume foods. Just briefly, I've been working in relating to Finnish and Nordic food systems, and perhaps the most mostly consumed plant protein sources are cereal grains, so wheat, rye, oat, and barley. Legumes can be grown in Nordic countries, and in Finland, particularly pea, faba bean, and lupine would be the choices that we should actually increase the production and both consumption. There are also been discussions about some other protein sources such as hemp and buckwheat, and even linseed and rapeseed. So plant protein sources have many beneficial nutritional compounds or contents. Most of all, they are, of course, good or moderate protein sources, so that making them important when we want to reduce animal-based foods. They are excellent sources of dietary fiber. They usually have quite a low content of fat, and the fat is of good quality in terms of fatty acid composition. They are rather low or moderate in energy content, which would be very important when we fight against obesity. And also we've got quite many vitamins and minerals. First of all, folate, that is the intake in Nordic countries is not actually to that level that is recommended in nutrition recommendations. There are also other vitamins and minerals and also phytochemicals that may have health effects. And just to mention carotenoids, vitamin K, and kalium, and yes, polyphenols. There might be some possible drawbacks. So even if the protein content is good, there is a limited amount of essential amino acids. It's well known that cereal grains are low in lysine, and legumes are low in sulfur-containing amino acids such as methionine. So we obviously need to think about how to combine these protein sources to achieve good amino acid composition. There are also anti-nutrients present, such as tannins, phytate, protease inhibitors that may interfere with the digestibility of not only protein but also some minerals such as iron and zinc and calcium. There are also, of course, differences in the micronutrient content between animal foods versus plant-based foods, and that may affect the intake of some crucial micronutrients such as vitamin B12, iodine, also iron and zinc. So, but what we don't know is that what actually will be the nutritional consequences in our populations, and if we take this dietary, I would say we need to take the shift towards more plant-based diets, but I think we really need to do research to understand what will happen to dietary intakes of nutrients, and whether there are possible drawbacks we need to deal with. So protein sources come with different composition of other nutrients, such as fats, carbohydrates, vitamins, and minerals. So when you change your protein sources in your diet, you also affect what else you get. And I'm actually not that much concerned about the amino acids as such, but more about the other nutrients. And you'll see perhaps why I think like I do. So let's start with the 2 randomized controlled trials we have done. And I'll first tell you something about the Skenopro trial, where we replaced animal-sourced proteins with plant-sourced proteins in a dose-dependent manner. Here we have had in both interventions healthy working-age subjects. In Skenopro, we had both women and men. The length of the intervention was 12 weeks, so 3 months, which is quite good or quite long. period, and then this was done in a parallel setting. We have the other trial called Beanman, where we concentrated on red and processed meat and replacing them with legume-based foods. This wasn't that big change as in the Skenopro trial. And here we concentrated on healthy working-age men. Why? Because at least in Finland, men are those who consume double the amount of red and processed meat compared to women. So we thought that, okay, let's see what happens with men. This was only 6 weeks and also done in a parallel setting. Then about the Skenopro trial, so we had 3 diets there. The first one representing the current Finnish diet, around 70% of proteins come from animal sources and 30% from plant-based sources, and that's mainly cereal grains. We started slicing off some of the animal proteins and replacing them with plant-based protein sources, such as legumes. Also, we had seeds and some sort of a all we could figure that could be where we could replace the animal sourced proteins. So, and the plant-based sources were, I would say, faba beans, pea, only some soy products, quinoa, seeds and nuts. And we decided to keep fish and egg consumption constant because I'm always afraid that if I meddle too much with omega-3 fatty acids, I end up having difficulties in interpreting the results. We had 2/3 of women and 1/3 of men, and ending up to 136 participants. This was pretty highly controlled trial, so we got 80%, or we gave 80% of energy sources as foods to participants. And here I must say we got most major Finnish companies involved, so they gave their products freely to the intervention. They had no role in planning the diet, the study design or interpreting the results. But I think industry needs to be involved if we want to make a change in our diets. We collected samples and collected food consumption. So how did we succeed? We aimed at keeping the protein intake constant, and that is the 70 energy %. We managed that quite well, particularly with the 50-50 group and the most plant-based group that is having only 30% of animal-based proteins and 70% of plant proteins. So there we didn't quite reach the target that was 50 energy % for proteins. And perhaps that shows that when we gave them pulses as such, so perhaps they weren't that eager to prepare those to foods. So that might be one reason. But I think that was pretty nice anyway, because we couldn't see any differences in carbohydrates or fat intake. We calculated essential amino acid intakes, and now I'm showing you these results using so-called box plots. And just to remind you that the line there is median, and the box represents 50% of the observations, and each of the whisker represents 25%. So when you look at, here we have lysine intake, it is of course highest in the most plant-based group. It decreases dose dependently when we move towards the most, So the intake is highest in the most animal-based group and decreases when we go towards the most plant-based group. the second amino acid or amino acid group most affected was sulfur-containing amino acids, and there you'll see the same dose-dependent decrease by the replacement. However, when we look at the compare the intakes to the requirement levels here, so you'll see that even in the most plant-based group, the intakes are about all right, particularly if we take into account the energy intake. So if you have a very low energy intake, you'll end up having pretty low amino acid intakes as well. So as far as you have sort of like in a range of normal energy intake, you will be fine with this replacement. We could see favorable changes in fat quality. So we will see a reduction in intakes of saturated fatty acids and cholesterol, and in line with that, an increase in intake of polyunsaturated fatty acids, and also a small but significant increase in N-6 unsaturated fatty acids. And this is now because we kept fish intake constant. This comes from alpha-linolenic acid. So obviously, if you consume rapeseed, as we do in Finland, you'll end up having even a little bit increase in omega-3 fatty acid intakes. So it's no surprise that when measuring blood cholesterol values, we can see a decrease both in total and LDL cholesterol. And I think this is sort of like very clear dose-dependent decrease. So no change in animal-based protein group, reduction in 50-50, and even bigger reduction in the most plant-based group. Fiber intake increased, and even if you'll see that even in the animal group, these were health-conscious volunteers. So even in the animal protein group, they had on average 25 grams per day of fiber. But the change or the replacement still increased the intake, and you'll get it, actually the median value is nearly 40, I think it's 38 grams per day in the most plant-based group. So we really can increase fiber intake significantly by changing the protein source. We've looked at some compounds in the stool that would indicate a risk for colorectal cancer, and we've measured so-called enricer compounds in the stool, and there you'll see like a dose-dependent decrease there by the replacement. So when we think, if we believe that enricer compounds may contribute to the higher risk of colorectal cancer, then we'll see like a clear decrease in the content in the stool by the replacement. So folate, as I said, legumes are supposed to be a good source of folate, and we will see a mild increase in the folate intake per day, but unfortunately we couldn't see any effect on the blood folate concentrations. This is now the baseline versus the endpoint. So what you see here is a 45 angle line, so meaning that nothing much happened. Of course, there's some variation, which would be perhaps interesting to look at later on. But in general, not much happening in folate status. Iron status has been very much discussed related to sustainable diets, and so we measured iron intake, and what we actually can see is an increase in iron. And where that comes from, there is, of course, a decrease in animal-derived iron, meaning that animal-derived iron here mostly means heme iron that is very well bioavailable. Of course, that's very well known, but we could see rather big increase in plant-derived iron intake that clearly probably compensates the higher bioavailability of animal-derived iron, because we couldn't see any effect on iron status markers. So we look at blood hemoglobin, and this is now once again baseline versus the end of the trial. And if there would be like a change, we would end up to different angle of the line, but it's mostly 45 degrees, so indicating nothing much happens. We looked at plasma ferritin levels, same story, nothing happens. And we look plasma transferrin receptor levels. There was something small going on, but really the p-value wasn't truly statistically significant. So we have 12 weeks of intervention, and the actually red blood cells should have changed. So we expected that in this time frame we should have seen something if something happens. So no change. So that was the good news. Now comes the bad news. So we looked at vitamin D and calcium intakes, and now this trial was done from January to June, and we said that the volunteers are not allowed to use any dietary supplements. So, of course, if you think vitamin D and Nordic countries, supplements may be needed, but these results need to be interpreted in the context that no supplements was used. When we look at the vitamin D intake, so actually none of the groups reach the recommendation only by dietary means. So they are all under the recommendation. Calcium intake decreases quite clearly by the replacement, which is clear because we reduce the dairy product consumption. So that explains this one. Here we'll see that the animal protein group does reach calcium recommendation. 50-50 is about 50-50 above and below, and majority of those in the plant-based group do not reach the recommendation. We looked at parathyroid hormone concentrations in the blood, and this is now baseline, and here are the dietary groups. So we could see that the most plant-based group had higher concentrations of parathyroid hormone regulating calcium metabolism and perhaps bone health in humans. And when we measured bone turnover markers, and here we can see that the plant protein group differs from the two as well. And this may mean that the bone turnover means that there's a higher turnover of rate in bone in the most plant-based group, which might indicate increased risk for bone fractures. And this is actually been shown in a Oxford cohort that regarding bone health, that there might be some problems in the most plant-based eating groups. Vitamin B12. This was, to me, sort of a bit of a surprise. Not perhaps that the intake decreased quite a lot by the replacement, but that actually the status marker decreased as well. So, and these were statistically highly significant. So we really need to think about vitamin B12, I think, when shifting towards more plant-based diets, and perhaps the supplements might be needed at some point. And just to remind you that the new Nordic nutrition recommendations have set the adequate intake to 4 micrograms per day. And here we have the old recommendation that was 2 micrograms per day. So if we take these 2 grams, the 50-50 group is still all right. but if we take the new recommendation, only the most animal protein-based group is fine. So certainly some measures needs to be thought here. And of course, this intervention was made in the time when the plant-based milks were not fortified. So now it would be really interesting to see what happens with the fortification. So if we use the plant-based milks fortified with vitamin B12, then whether that would sort of correct this not so nice result. Iodine intake did decrease by replacing animal proteins with plant-based proteins, and that seems to pretty much dose dependent as well. We measured urine iodine, which is like the golden standard of iodine status. So there, once again, we'll see the dose-dependent reduction when removing towards more plant-based diets. And this is, of course, another issue, I would say, we need to think at population level. We actually have calculated the climate impact of the trial diets, and this is no surprise, sorry, but just to say that this is perhaps the first time when climate impact has calculated for every volunteer on real diets. So usually those are the LCA calculations are sort of like done for certain model diets, but this is now what happens in real life, and we can see that if we calculate CO2 equivalents per food, yes, there is a reduction from animal 50-50 to most plant-based diet. The same if we adjust per energy intake, though it's not as drastic, and if we adjust it per protein intake, then the trend is there as well still. So conclusions based on the Scanopro trial, replacing animal-derived proteins dose-dependently with plant-based proteins led to decrease in the intake of saturated fatty acids and cholesterol, increase in the intake of unsaturated fatty acids, both N-6 and N-3, which was accompanied by a decrease in total and LDL cholesterol, and hopefully reflecting to reduced risk for cardiovascular diseases. We could see increase in fiber intake and decrease in stool and natrium compounds, indicating better gut health and hopefully a reduced risk for colorectal cancer. But then we could see a decrease in vitamin D and calcium intakes and an increase in parathyroid hormone concentrations, accompanied by an increase in bone turnover marker, suggesting higher risk for bone fractures. And I would say that attention needs to be paid on the adequate intakes of vitamin B12 and iodine, and perhaps iron is not that much a problem. I'd actually like to refer to a systematic review that was published last year, and it looked at all the studies that have been published on what happens on micronutrient intakes when removing towards more plant-based diets. And this, I think, included something like 50 population studies and one RCT, which was ours. And here you can see that the increase in iron and folate intake was actually seen in most of those studies as well. And a decrease in zinc, calcium, and vitamin B12 and vitamin D intakes was also seen. So very much in line what we saw in our real-life randomized control trial. Then the Beeman trial. So this was sort of a more subtle change. So we only controlled red and processed meat intake and replaced them with legume and meat. With legume foods, and we took the 760 grams per week of red and processed meat consumption by Finnish men as the control point. That represents 25% of protein intake, and then we reduced the red meat consumption to 200 grams per week, representing the planetary health diet, the upper limit in planetary health diet, and the extra protein was then replaced by legume foods, providing 20% of the protein intake. And most of these foods were based on pea and faba bean, because we were thinking that it needs to be something that we can produce in Finland. So we have here 102 participants, 51 per group, and surprisingly no dropouts. This was actually done during corona pandemics, and the men seemed to have nothing else to do than participate to our trial. So I was a bit, I couldn't believe my eyes because, at the end that no dropouts. The mean age, these are healthy working age men, mean age 38 years. There were no differences between the groups at baseline. We all once again collected blood, urine, and fecal samples and collected the food records. Here you'll see some of the foods we gave. So these are the the foods given to meat group. There are all sorts of like meat as such, but also sausages, and this is sort of liver paste, which actually some didn't like it at all, but because it's taken our Finn diet results, it's actually consumed surprisingly much. And these are the foods given to the legume group. So we did give them some of the meats, food, so both processed and meat as such. And then we had some of the legume foods here. So legumes fresh frozen, in fresh frozen form, in different kind of plant-based pasties, and then this one is can be used as a minced meat. So and that was all they needed to do: eat what they were given, and otherwise not change their diet. What we could see that there was no differences in energy yielding, nutrient intakes, which I was very happy because we wanted this to keep constant, because we really wanted to see what's the effect on other things. So we've looked at amino acid intakes, calcium intakes, vitamin D intakes, those that were perhaps changed in the Skenopro trial. Here we actually couldn't see much of an effect. So this graph is about amino acids, and there were a little bit, some decreases in for example, in histidine and in methionine in the legume group compared to meat group, and some increases in amino acids like alanine, arginine in the legume group. But none of these were sort of close to not sufficient intakes of any amino acids. So just sort of like changes, but not probably having much clinical meaning. Of course, that can be debated, but because amino acid intakes haven't been reported much. So I think we are lacking in protein nutrition research compared to, let's say, what we know about dietary fats. And no differences between calcium, vitamin D, nor the biomarkers of bone health, which I think this is good news. What we could see is, once again, decrease in saturated fatty intakes. And here now we have these light colors indicate the baseline, and what you need to look at is the orange compared to dark green. So there's a increase in polyunsaturated fatty acid intakes, and once again also in the N-3, which is linolenic acid. What happens with total cholesterol? We actually could see here the same as in Skenoprot, so there were decrease in the bean group, both in total and LDL cholesterol. Nothing much, at least in statistically significant way, was happening to LDL cholesterol, a small decrease in the legume group. So this seems to happen quite, how I would say, when we replace animal-based foods with plant-based foods in Finnish dietary context, it seems that the blood lipids are affected. We could see here an increase in fiber intake in the same way as in Skenoprot, and we could see actually quite dramatic decrease in the enriched compounds in the stool. So our hypothesis is that enriched compounds probably are one of those compounds that may mediate the effect of red meat, high red meat consumption to colorectal cancer risk, because they are known to be carcinogenic in animal models. Iron, this is of course once again interesting because red meat is a good source of heme iron. We could see the same thing here as in Skenoprot trial, so there is a decrease in animal-derived iron and an increase in plant-derived iron. And actually, we looked at the iron content of many of the legume foods, and it seems to be surprisingly high. So we needed actually even double check some of the iron contents in those, I would say, processed plant-based foods we gave, and it really seems that they somehow, if you use actually protein concentrates to make foods, protein concentrates also concentrate iron. So that might be the reason for that. We could see a decrease in vitamin B12 intake and also in the status, but these are nothing so dramatic as in Skenoprot trial. And of course, men are not a vulnerable group in a way that they eat more than women usually, meaning that their intakes of nutrients are higher by default because they eat more food. So here we concluded that this might not be a problem because both the adequate intake and the blood values for hollow TC are, I would say, most of the values are above the critical recommendations. So at least in this short time, no problem. Iodine intake did decrease slightly, and it is statistically significant, even though the in quantitative means it's not that drastic. And we don't know if this is a problem or not in the long run. So conclusions based on the Beanman trial, when reducing red and processed meat intake from 760 grams per week to 200 grams per week and replacing the subsequent protein intake with non-soy legume foods in the diet in Finnish men led to no changes in macronutrient intakes, small differences in few amino acid intakes, but otherwise intakes are adequate. Dietary fat quality improved and total and LDL cholesterol decreased. Fiber intake increased and stool enriched compounds decreased. We could see statistically significant decreases in vitamin B12 and iodine intakes and actually in the status, but we don't know if this is clinically significant. The changes seemed pretty small to our eyes. Overall, we feel that this moderate replacement seems to be feasible to follow and it's nutritionally safe. what I think needs to be discussed is that if we take if we make a large dietary change, so what happens at population level? So really we need to hopefully have those studies that do these comparisons at population level, and then we need to look at if we need to take some actions to secure, let's say, vitamin B12 and iodine intake. And I'm actually not worried in vegans as such, because they very well know that they need to take B12 as a tablet or supplement. But those, let's say, flexitarian people who perhaps sort of a shift towards more plant-based diet and might be actually quite close to vegan diet, and then don't realize that they might have problems with vitamin B12 and iodine, then I think that sort of a target group is important to take into account. And of course, the long-term consequences, I think vitamin B12 is something we really need to look at, because when I was a student, I was taught that, hey, you only need like a very tiny amount of vitamin B12 and it sort of lasts forever. I think it doesn't. So that's the work where we need to work further. These are the people who have taken part. And actually what I'd like to you to think about a little bit is this line, which I'm proud because I'm sort of have created this in one project. So when we go from the mostly animal-based protein diets towards mostly plant-based protein diets, it's clear that the climate impact decreases, but we then might end up with different problems. So in this end, we have some good things, so like high quality protein, sufficient amount of essential amino acids, bioavailable heme iron, and so forth. Risks are related to fat quality, high intake of saturated fat, low intake of fiber, etc. And when we go towards this end, we really have the quality of fatty acids improves. We've got more fiber, which I really believe is a very important thing regarding our health. We've got folate, phenolic compounds, etc. But then we might have some problems with vitamins such as B12 and calcium and with iodine. So thank you for your attention.
Thank you, Anna Maria, for this wonderful overview. Any questions from the floor, from the hall? A thing which I would like to articulate in this frame as well is that if we think of plant-based proteins, we are very often concerned that the uptake is less and could be in some groups not enough, but we are often neglecting the effect of the malabsorbed protein, which enters the colon and then is fermented. And in Penn Sweden, we take a lot of attention to that, and see also, for example, that if you increase the plant-based protein intake quite a lot, that there is a subgroup who develops a higher level of inflammation in the gut, still within the normal value, but it is absolutely significantly go. Any reflections to that?
Yes.
The other sort of disadvantage.
Yeah, I think, yes, that's something I've been thinking as well. I haven't studied it. Happy to hear that you have done it. Yes, I think that might be a problem. I don't know if we can, I'm always thinking like solutions, you know. So if, perhaps it's too early to say how detrimental that is. Of course, inflammation is a risk factor for chronic diseases, but then we have on the other, let's say, the balancing part is the fibers and actually the interaction of proteins and fibers should be studied quite a lot. And I wouldn't be surprised if there is sort of like an optimal ratio of animal and plant-based proteins in a diet, because obviously it benefits our health if we have some of the animal protein there still remaining. But so what would be like the optimal ratio of those two? I don't know, and I think we need to study that.
That's exactly what we are doing with the hypothesis that increasing dietary fiber changed the microenvironment in the gut and could actually diminish the detrimental effects of protein malabsorption. And we have indications that it is true. Yeah.
Okay.
Any other questions? I look around. Yeah.
Yes. Thank you for a very nice presentation, and I think the results of the high iron levels of plant food is very promising, and also that the iron station doesn't seem to be affected. This is good, and I think we still maybe need to be aware of that for some groups like children and adolescents where the needs are higher. There's still possibility that we need to look at it, and possibly also to make more long-term studies, since I know that although these 12 weeks may be enough to see an effect, but it takes quite long to rebuild depleted iron stores, and maybe it's the same to restore them. I think it looks promising.
Yeah. Thank you for the comment. Actually, I must now emphasize that we are working with what we have been working with working age population, which is the least vulnerable. So, and I was thinking whether I'll take the protein bioavailability study we have done here in our group. It's of course well known that protein bioavailability and digestibility is less in plant-based proteins, and I don't think it's a problem with healthy adults, but it can be a problem in elderly people, and of course we don't know about children. So the same way as iron and vitamin D, the vulnerable groups should be studied.
So I meant iron.
You meant iron, yes. But but iron, vitamin B12, iodine, I think they very much go in hand. Let's say when we think about the growth of our children.
There was a question there, last question. Yeah.
Yeah, just a short one. Thanks for a very inspiring presentation. And I just had a short question about the Seneprot, the first study with plant-based drinks, given that I come from Oatly. But was those products, were they fortified or were they not fortified?
They were not fortified, at least. So we actually gave some plant-based milks, but those were not fortified because that didn't happen at that time yet. That came later on. If you ask about some of the, let's say, processed plant-based products, those, I think some of them might have been fortified. I'm not quite sure. Can't remember it exactly.
So we would like to thank you very much again.
Thank you.
Welcome everybody to this second session. I'm Maud Langton. I'm professor at SLU in food science, specialized in structure and properties.
And I'm Anna Ström, and you saw me before, I forgot to say my name. And I'm based at Chalmers and also co-director of Pan Sweden together with Robert.
We are happy today to introduce Professor Kati Katina from Helsinki University. Kati has also been guest professor in my group, so I know her. And you know that processing has a huge effect on the food. We're happy to listen to your experience.
Thank you. Okay, first of all, thank you very much for invitation to give a talk here today, and especially after this exciting and wonderful presentation heard today. And my task is to give you a little bit different perspective in this story of what shift we need to do, and what is the challenges from technological perspective, food tech perspective to this story. So no more justification needed. It's clear what we need to do with shift due to sustainability, health and ethical reasons. However, when we are thinking about plant-based foods and plant-based raw materials, there are challenges which already nutritional ones we heard. So there are nutritional challenges, and there are technological challenges, and there are sensory challenges. So thinking about food, I'm not talking about the nutritional challenges, you already heard about them. But if you think that what kind of plant-based foods and plant-based proteins we consume, there are quite a variety of options already. If you think that the traditional way to eat, for example, peas is pea soup, I don't think that is a future food, what we are going to consume in huge amounts. So there are some processing related challenges in plant-based proteins and plant-based foods in general. And some of the issues what we have is that plant proteins are really different from technology perspective. They have very many of them have low solubility, and actually they have rather poor technofunctional properties. And maybe one of the most severe challenges in many of them, that they have off flavors. They are not very good tasting. And their structure formation properties and texture formation properties are also often quite challenging. But we have a solution. Food technology can really alter and solve these issues. But there are all of these aspects can be somewhat corrected with the means of food technology. And I wish I would have even more time, but because I have this 40 minutes, I will focus actually two challenges. One is structure formation in plant-based foods, and I take the case of extruded meat alternatives. And second challenge I'm talking about is flavor of plant-based foods, and where I'm presenting some recent results done in Pajari in my group. So, structure formation in plant-based foods. Case high moisture extruded meat alternatives. And I specifically ask in presentation of Paul that how was actually the proteins and fibers, how they were consumed. And typically when we think how we nowadays consume plant-based protein and foods, they are often very processed. They are quite heavily processed, and that may or may not change also other properties like nutritional properties. And as much as we would want to make this change as quickly as possible to eat more plant-based proteins and foods, there are some challenges. And unfortunately, we had maybe during last 10 years this hype that we have had a nice increase in plant-based options, also plant-based meat alternatives. But unfortunately, during last 2 years, I can't get the pointer right, but you see it, that there has been decline. That plant-based meat decreased 28% during last 2 years. And this is a USA market, and unfortunately recent review says that the same story goes for Europe. Both the dollar sales and unit sales has decreased. And main reason given in recent reviews is that the sensory properties are not good enough. Both the taste and texture are inferior. And the situation is such that in one study, consumers tried meat alternatives only one time, and then never again. So that was absolute no after trying it one time. And then the sensory quality has to be rather low. So something has to be still done, that we would get this increase in the curves and not really that we have a declining curves. Of course, there are other players in the picture, like we have had Ukrainian war, and also we have had COVID, and people, when they are choosing the food, there are many factors influencing a food choice. It's not only that you want to save the planet. For example, if you are living uncertain times, you can go back easily to meatballs and potatoes because that is a safe option. That gives you security. So it's much more complicated than that we would only want to save the planet. What are the challenges in meat alternatives? Actually, there are a lot of them. We need to really combine number of things to get successful products. First of all, it starts with the protein selection. It's funny when we are talking about plant proteins and there are at least 20 different options or more. There is no such thing as plant protein. There are individual plant-based proteins and all of them are different and they behave differently in the processing. Then we have number of processes to choose to create a texture which would please the consumers, that they would actually buy the product. And the product itself, it has to have a good taste and texture. It cannot be very expensive, preferably clean label. People don't want to eat additives. And there should be a diversity in plant-based protein foods. And nutritionally, of course, they should be good. And that is also a challenge, as we heard. And of course, the process itself should also be cost efficient and didn't cause any more sort of a burden for environment or the planet. So all of these factors actually should be combined to have a successful plant-based and plant protein-based foods. So quite a lot to solve. And in an ideal world, we would eat different type of structures. But unfortunately, especially in this transition phase, companies have much focused, and I think it's necessity and consumer preference, but we are trying to mimic the meat. And that's really difficult to mimic. And if you think about the meat structure, it has a very, especially whole meat, thinking about steak type of meat, it has very, very complex hierarchy of muscle tissue, adipose tissue, and connective tissue surrounding the muscle fibers. And because it's very complex structure, it's difficult to understand the physical, chemical, and functional properties, and very difficult to mimic. And plant proteins are completely different to animal proteins. We already hear that they are structurally very different, and we need to completely alter them to have meat-like fibers and get the texture that would resemble the meat. So that is a challenge itself, not very easy to do. And if you think what is now available or have been available for a while to create meat alternatives, there are a number of options. Where is that you use plant-based raw materials and you use some kind of extrusion technology to produce meat-type foods, texture-wise. And that is already, technology readiness level is also important. This is a commercial technology. There are a lot of products produced with this technology. And then there are more sort of research and development level technologies that you have these cell-produced meats or you have these microbial protein-based foods, and they are still very expensive and they are not commercially available, the masses. And especially, I think it was in the first presentation that foods also should be available and affordable. And if you think that we would have a fancy cell technology who no one can afford, when it doesn't save the planet or solve this transition in our diet. And the most common one, what is now commercially available, and what you can buy from a supermarket is extruded meat alternatives. And that's why I'm talking about them and challenges related to this technology. And if you think that you take your plant protein and make a structure and sensory properties somewhat similar what you have in meat to appeal the consumers, you need quite a lot. You need plant proteins, and you need processing to modify these plant proteins to concentrates and isolates, but you can produce meat alternatives. Then you typically need flavoring agents to create nice appealing flavor, sometimes also mask the off flavors. You need texturizing agent, but you can get these fibrous structures. You need other additives, maybe nutrients, and then you need extrusion technology, structuration technology to create these meat-like structures. And you can forget, in a way, naturalness when you are creating these type of foods. But you need a lot of different type of ingredients and intensive processing technology to create meat-type structures. And of course, if you start from raw materials, we have a lot to choose. And most of the efforts have been sort of focusing on legumes and cereals. And even in legumes and cereals, you have really plenty of options to choose. And this is heavy table, and we don't spend a lot of time it, but you see that even in most common ones, and also observe here that how much they are globally produced. As much as we want to use algae or some novel plant-based proteins, you also have to consider that are they globally available, how much they are cultivated. Because, I go too fast, if we really think that what crops are now available to feed the world, there are actually 3 crops which are cultivated in massive amounts. And that is wheat, rice and maize. And from the pulses is actually soya, which is available globally. And everything else is minor crops. So if you think the plant proteins, you also need to consider what is available to be eaten globally. And technologically, and what is technologically maybe not that much thought when you think about the nutrition, is that they are all different. They have different water absorption, they have emulsification, gelling capabilities, and these differences are based that the protein composition and behavior are different. So it's quite a big generalization to talk about the plant proteins and make a thing that plant protein is more available or not. It completely depends what kind of plant protein you are talking about, and especially how it is processed. And this is one nice picture about the plant proteins. Most plant proteins have really a globular type of structure, which is this is soya glycine hexamer as an example. And what does this mean? That it actually have quite complex multimer structure. There is numerous different type of proteins held together by physical and chemical bonds. And this is now a native protein before any processing. And as such, these lack functional properties that we could have a textures similar, for example, like meat. And in order to really create sensory textures or structures which would be somewhat similar than meat, you have to do really intensive processing. And also the proteins are need to be quite tailored to obtain certain specific properties that you can do high moisture extrusion successfully to get these fibrous structures. Proteins have to have, high solubility, high water holding capacity. The different protein structures like 7S and 11S protein structures have to be in optimal ratio. They have to be right ratio of hydrophilic and hydrophobic areas. There have to be disulfide bonds to be broken down and rebuilt. There have to be a melting enthalpies which actually are matching to the energies used in extrusion. And they have to have certain specific viscoelastic properties, but they have to have this non-Newtonian fluid behavior and have shear-trimming properties that you can have a successful extrusion. So quite a lot of demands for proteins for processing. And what actually happens in extrusion? Well, you can create meat-like textures. Then you first, oops, I'm not very good with this one. Sorry. When you do the extrusion, you actually can do the structures, but you have to really melt down the protein and restructurize it. And what happens is that you have a native protein, globular proteins, and what you do, you need to unfold them during the searing of extrusion. Then you need to, these unfold proteins need to, associate and aggregate when the temperature increases and there is more shear increased. And finally, when the process continues, you have complete rearrangement of these molecules. So you break down them completely and then you rebuild the protein. And if that is happening in an optimal way, you have an optimal rearranged molecules, you will have these fibrous structures created from really globular proteins. So it's massive change what we are doing to the proteins. And what does this mean? But it is possible to mimic the meat, and you can buy already like a chicken meat type meat alternatives. But the plant proteins have to have properties to match to the extrusion process, and each time when you have new mix of raw materials or different type of proteins, you have to do the process optimization in a raw material specific manner, protein specific manner. And still the current meat analogues are not good enough. There are flavor challenges, meatiness, juiciness, many sensory attributes are still not according to consumer liking. Nutritional quality, I just discussed with Anne-Maria at airport in the morning. Do we really know what we are doing to the sensory quality? That is sort of a nutritional quality, sorry. I don't think so. But there is a lot to be done in the future. But if we go with that route, that we want to have meat-like textures, for example, how do we optimize texture and nutritional quality at the same time? I think we're not even, maybe the audience can comment this later on, I don't think there are even many studies, at least in vivo, who has really studied intensively processed plant proteins. But what do we change there? And now this discussion which has come up in several conferences, ultra-processed food. And as a processing person and food technology person, I'm certainly not against processing. I think it's must to have a good quality food. But if you think high moisture extrusion, to be honest, it is highly processed. We are completely changing molecular arrangement of proteins, and what else we are changing, we are not even knowing for sure. And of course, change can be also positive. Maybe the plant proteins are behaving better after certain type of extrusion, we don't know. But I think it's really interesting research field. I think it's next 50 years we can study this. Anyway, now the bigger challenge, taste challenge. And the lady here looks angry, and she has reason to look angry. Most plant-based protein sources, soya, faba bean, and peas, have severe flavor challenges. And what my group has especially studied is bitterness. There are other ones, speeniness, aftertaste, astringency, But bitterness is typical for many beans, and it's difficult to solve. First of all, bitterness has very complex flavor chemistry. There are dozens of different bitter tasting compounds in plants. They exist together and they cause bitterness. Then individual differences are huge. My sensation of bitterness is different than somebody else's sensation of bitterness. So even sensing the bitterness varies a lot from one person to another, and also from one culture to another. And the bitterness is also historically has been a warning signal. When our ancestors were grabbing new plants, when they were wandering in forest, they tasted, and if it was bitter, it was considered as a warning signal. This might be toxic. And when it was bit off. So all of these are making this picture quite challenging to solve. And I have against faba beans. And if you think faba beans, we dehull them, we mill them, then we fractionate them. We can fractionate them to protein concentrate, up to 60% protein. We can use flour, it's already have 30% of protein. Or we can do wet extraction to get the protein isolate, which has about 90% of protein. So nice ingredients. And we can use them in number of food applications. We can use them in bakery products, bread, pasta, etc., biscuits. We can use them from dairy alternatives, nice options available, and also in meat alternatives. And we can get nice nutritional properties, high in protein, rich in fiber, not inulin, some other fibers, and they can be considered healthy. And of course, this is a sustainable way to use them, and it's sort of texture-wise, it's fine. But then comes the issue of flavor. But is it flavor really appealing enough to that consumer would really buy these products? Based on the current data of consumption, it is not. I hope this situation, of course, there are a lot of research efforts here, and the picture is changing, but currently it seems that these are not good enough to be accepted by consumers. And of course, when we are thinking about industry doing new product development, they have to take into account if consumer like the taste and texture, otherwise nobody buys them. And flavor is a nice thing to study. It's so complex. It never sort of gives up for surprising you. And if you think flavor itself, first of all, we have smell perception. We have volatile compounds, which are reacting with olfactory receptors and creating actually sensation of smell. When we have same with the taste, we have non-volatile compounds. They react with taste receptors and give the basic taste: sweet, salty, bitter, sour, umami. But when we have other things happening there, that physical properties, texture and temperature are influencing the sensation, sort of the somatosensory perception, and also some volatile and non-volatile compounds react with sensory nerve endings, and they can give a chemostatic perception. So this is really complicated already now. More complicated it is when we think about food structures. That if we have exactly same type of chemical compounds in different type of foods, it is sensed differently. Because food matrix influences how we are sensing different flavor compounds. And these chemical, biochemical, and physical properties of compounds, and they are interacting with different components in foods, like starch, lipids, other proteins, for example. And these interactions are influencing how the sensation is actually happening in the mouth. And also, we have individual differences, not only for bitterness, but we have cultural, psychological, genetic differences, how we actually sense different flavors. And of course, there are environmental factors as well. But if we are tired or if we are eating with family or alone, that influence. And this is a recent study was done in my group by Fabio Tuccillo, who is defended this year. And all of these concerns, if you ask in a questionnaire from people, but what is your most important thing to choose, for example, plant protein-based foods, they might say that it's healthy and it's ethically correct and I want to save the planet. But when it's really come to the food choice motives, it's sensory appeal. And liking, do we like food or not? That is the main driver of consumption. And all of these other reasons are secondary. Ethical concerns, weight control, price, convenience and health are secondary to the liking. It's really that simple in Finland. I think it's also in other countries. But if you like or not the food, you sort of choose and buy it. And in this PhD work of Fabio, we studied faba beans really in detail. We first of all studied consumer acceptance, liking of smell and taste, overall liking, willingness to use faba bean ingredients, and how these were linked to the consumer characteristics, sort of different properties of consumer groups. Then we studied the sensory perception of different ingredients and foods made of them, faba bean based. We characterized the odor, taste and flavor of faba bean ingredients, and we tried to link them to the liking or non-liking. And then we studied ingredients, unheated ingredients as paste, and when we heat treated them or did the wet extrusion, we examined that how actually the sensory properties are changing when we do the food processing. And finally, we identified with targeted and non-targeted analytical approaches the potential flavor compounds behind these different sensory properties. And we had these commercial raw materials. We have faba bean flour, 2 faba bean protein concentrates, one from Canada, from Finland, and 1 protein isolate from Canada. And we studied all of these aspects identified in aims. And we studied them as a pastes, unheat-treated paste, and we did the high moisture extrusion. And the main results wasn't very promising. Nobody liked them. I think Fabio hoped that the results wouldn't be that sort of dramatic, but whether you are vegan or flexitarian, whether you have high aims to save the planet, these ingredients, the red color is not liked, and darker the red color is very much unliked. And green color is that you like them, and dark green that you like them very much. So you see that nobody liked them. And that was a very, very clear result. And acceptance was extremely low, and willingness to use was extremely low. And these are commercial ingredients. These are not sort of experimental ingredients. And of course, the situation when we did this study was a few years ago, and I do hope that there are some development done now. But at least when we 6 months ago at SLU tasted new faba bean concentrate, it was really bad. So maybe some product development would be needed still, I would say. But luckily, these are not the same. Depending on raw material you select, is it the flour, protein concentrates, or protein isolate? You remember this was 30% protein, this is 60, and this is 90. They are quite different sensory-wise. So flour was sweety, fruity, even beany somewhat. Protein concentrates were actually burst. They were astringent and bitter, and protein isolate has more sort of wider nutritional sensory properties. So nutty, earthy, cheesy, even cooked bean flavors were present there. So they are different, which is actually good news for product development. And then we connected the liking and disliking, and it was really the bitterness which dominated the picture. But if the ingredient and food is bitter, that was the main driver, is disliking, and everything else was a secondary. And this other type of sensory properties like sweety and cheesy and fruity and nutty were more sort of linked to the liking. What about processing? These were the paste, what we tasted, and when we did wet extrusion. And you know that the proteins, they change a lot, and there are high temperature, high shear. So how did the flavor change? Well, it of course changed a bit. Pea odor and pea flavor changed to the cooked pea odor and cooked pea flavor, and some cereal odor was changed to the cooked cereal flavor, etc. But unfortunately, the negative ones, like bitterness, that didn't change. If a raw material had bitter characteristics in paste form, it remained also after wet extrusion. And also the dryness of mouth, which an astringency type of characteristics, they remained after the processing. So we didn't get rid of them with the processing, this type of processing, what wet extrusion does. And volatile compounds, we found different type of volatile compounds, and lipids were linked to the odor intensity, cereal flavor, and off flavors. So there was different type of fatty acids, aldehydes, terpenes, ketones, and carboxylic acids, which were linked to these odor intensity, cereal flavor, and off flavors. And similar way, we had non-volatile compounds were linked to the taste intensity, bitterness, dryness of the mouth. And the compounds which were found is in this study, there's the ginger glucolipid, and quite interestingly, convisin and visin, which are the main antinutrients in faba beans, they were also linked to especially the bitterness characteristics of these ingredients and exudates. And then there was some amino acids like tryptophan, lysine, and phenylalanine, which were also linked to these characteristics and some other compounds. So there was sort of a clear sort of a potential flavor compounds linked to these sensory characteristics. So this is a challenge, not obstacle, to use faba bean. I'm not giving up. I hope you are not either. It's a good crop. Oh, I'm using my time. We can do plant breeding to develop new varieties which don't have these flavor problems. We can tailor the processing to make more neutral concentrates and isolates. We can use processing techniques like fermentation and enzymatic treatments. We can use masking, not just to put a chili sauce, there are other type of masking to solve these issues. And I give you only very quick example what we have done. We have done a certain type of bioprocessing, actually with lactic acid bacteria fermentation with specific enzyme activities, and we have created the exopolysaccharide, in this case it was dextran, which can be used in a number of different food categories, bread, dairy alternatives, meat alternatives, and what it does for flavor. This is a faba bean slurry control without anything, and if we put the dextran there, either enzymatically produced or optimally fermented, we can reduce the bitterness. And also traditional fermentation, which is more acidic, reduce the bitterness, but very strong acidic characteristics as well. So this is only one example of technological solution. We can manage these options, challenges with the targeted processing technologies and targeted food technology approach. So I'm hopeful we will make it together. I hope you believe me and don't be depressed. One day we can manage the texture and flavor of all plant-based proteins. And we did this also in extrusion, and we had a nice texture and flavor also in extrudate, so it does work. So there are solutions. But I'm lacking my time now, time is up, so I just some work done in my group related to that. It has been an interesting journey, challenging one, but interesting. And I like to thank a big team behind of what I showed. Thank you for your attention. And the clock was actually not working, that's why I didn't know the time.
Thank you. Thank you very much for a nice presentation. Questions? Oh, Johan?
Can you say anything about the energy cost of the extrusion process versus traditional cooking of beans?
I think it's much higher.
Yes, and then you have to cook them again and prepare them. So it's really
Yes, it's a sort of if you are trying to mimic this, create this like textures, you should sort of think not only about raw material, but the processing you do, and it's also the other type of processing that you should consider the energy intake.
It's huge.
Yes, you know even better.
And
than
you
me.
hinted on that this trying to resemble meat is maybe a sort of dead end in reality, that you should perhaps go back to more traditional.
Pea soup.
Yeah, pea soup. I like pea soup.
Anna?
I also like pea soup actually, but maybe not all the days. So my question is related to, so you saw very nicely that if you do fermentation with bacteria producing dextran, you saw a reduction in bitterness. So I wonder, is that related to just dextran being a polysaccharide? Could we just add any polysaccharide there and not do the fermentation? Or
Well, there was enzymatically produced dextran, so there was no fermentation.
okay.
So theoretically, you can produce dextran and throw it in, and it should work.
Yes.
If it's the right molecular weight and structure.
If I add arabinoxylan, for example, or another polysaccharide, that also, I mean, the dextran makes it quite thick. So
Yes.
it's sort of the thickness.
Dextran is not
that
very
is
viscous, actually.
Not so much.
Yes.
But if I add guar instead.
Other polysaccharides are promising as well. Not only dextran, it was only example.
Thank you.
Yeah.
One more question, maybe? Anne-Marie?
Yes, the decrease in bitterness, was that to an acceptable level, or you have?
Well, it was 7 and it was 3 and 5. 5 is sort of acceptable. Yes, it was acceptable. It was clearly reduced, but not gone completely. There was sort of very slight bitterness still, but it's individual, so my sensation is probably different than yours.
Mm-hmm. Thank you once again.
Thank you.
Lots of possibilities with processing.
Yes.
Yeah, thank you.
Thank you so much.
Thank you.
And with that, we are very, very happy to introduce Professor Bettina Wolf. And you are from University of Birmingham. And we started today discussing the importance of the industry being involved. And Bettina, you have experience from both industrial research and academic research. And you will continue to discuss what we can do with processing.
Yes, I will. Thank you very much, Anna, for the kind words, and we know each other from our time in industry, right? I feel this is a very different type of presentation here, so please, apologize if that's a problem. So I would like to talk about processing of plant protein-based food microstructures. So I've always been interested in creating food microstructures through process. So now do this, okay? Before I start, I would just like to get rid of acknowledgements. I'm showing data that were produced by PhD students and postdocs, technicians, etc., and colleagues at the University of Birmingham, colleagues at the University of Melbourne who co-supervised one of the other people who used to be a PhD student funded by a joint scheme, which is really quite interesting, and I would also like to thank Hura in Spain for funding, and my collaborator Sandra Hill from Nottingham, who is on a project with me funded by the Good Food Institute, which unfortunately finishes soon. So we haven't really come to our answers how to create textures that resemble muscle meat a bit more than just extrusion processing, because we actually found more. We opened up a whole load of questions and found rarely any answers. Anyway, so such is life, but that's fun as well. So as I said, I'm interested in food microstructure generation, and here's just a list of the different microstructures we find in our foods, going from emulsions all the way down to gel networks, which is probably what we should be talking about when we have, for example, plant-based meat analogues. Why are we interested in food microstructures? Why do we need all of these microstructures here? They impart actually the physical properties of my product. So if I have a certain flow behavior or viscosity of a product, I can feel it in my mouth. So this is what the consumer would do. I would call it maybe thickness and mouthfeel then. But all of these properties are related to the microstructures. They don't come just out of nowhere. And also textures. These physical properties are also important for processing, of course. Kati just mentioned we need shear thinning materials to successfully run an extruder. So to create food microstructures, we need processes, unless, of course, we're talking about natural food microstructures. So we often try to mimic what nature can produce by itself. And that's interesting in itself as well. And these also have, of course, then certain feel and physical properties. One of these natural food microstructures comes up a little bit later in my talk, which would be oil bodies that we find in plants, which are natural emulsions, basically. But in any case, so we need processes. These processes, as we heard, need ingredients. There's primary processes to produce ingredients from commodities, such as fava bean. And sometimes we might be wanting to use the commodities directly in processes, and they also impact microstructure. So moving on, my recent interest in plant-based proteins, and I've also only entered this field a few years ago, so I'm not really a protein person, but more interested from a microstructure point of view. And we really set ourselves the challenge to use our microstructure creation expertise to create something that would hopefully maybe look like a whole meat cut at some point. Okay, so as we already saw from Kati, we need plant protein, we need the protein source, we need, here I call it binder, so we need the processing aids, etc., to create the structures. And there is also maybe non-binder additives that are there for nutritional purposes or flavor, etc. So we submit these ingredients, we expose them to a structuring process, for example, high moisture extrusion, as Kati just mentioned. We create a fibrous texture, we get the texture profile. This is really weird here. We create the texture profile that then the consumer hopefully likes or doesn't like, and we also have a nutritional value. As Kati just mentioned, there's only very few references in literature that look at the impact of, for example, high moisture extrusion processing on plant proteins on the nutritional value and everything that means, nutrition, bioavailability, absorption, etc. There is a lot more literature on texture creation. And that, of course, is a big gap that needs to be filled. So, as I said, and we saw this already, this is a massive challenge to reproduce what we see in the moment, and Kati showed bigger images of this. This is a typical extrudate, as well as Kati already showed. So here the extrudate is sort of broken open, torn open in the direction of the extrusion process, and we can see these fibrous textures, which actually you can't see if you would a knife and cut the material, and that is also quite important. If you cut these things, you can't see those textures. You have to rip it open to see them. And this image here, the grey one, is a microcomputer tomographer, X-ray computer tomograph of a freeze-dried of this freeze-dried extrudate. So I stole this image from literature, and we can see it's a very complex composition here. There's 3 different protein sources, and this indicates a processing temperature here. So in addition to extrusion processing, like high moisture or even low moisture extrusion processing, other top-down technologies to create fibrous textures include shear cell technology, which uses much lower processing stresses and temperatures than extrusion processing, so we don't have the mechanical forces of the co-rotating or intermeshing screws in an extruder. And then there's also freeze structuring as a top-down technology to create fibrous textures. And here's a list of bottom-up technologies. I mean, we're all probably familiar with 3D printing, which on a gastronomic scale, etc., is used. And there's also wet spinning and electrospinning. So what I would like to talk about is some work from my group on quinoa protein isolate. I then would like to talk a little bit about our recent doubling with freeze structuring to create fibrous textures. And if time permits, I would like to show just a couple of slides on high moisture extrusion as well. Okay. So starting with microstructure engineering of quinoa protein isolate. So here we started actually off wanting to do something different with quinoa protein isolate. We wanted to choose an approach that's called phase separation to create meat-like textures, which didn't work because of the specific material properties of globular plant proteins. That was when I was very, very naive, still working with plant proteins. So the student then went on to look, how can we improve the technofunctional properties of this particular material to create structures and gels. The choice of quinoa was made by the student. The results are certainly translatable, even though we haven't demonstrated that to other plant protein isolate, at least in principle. So we've looked at changing the wet extraction process. And we then also looked, and I think this is really important for industrial application here, how do the technofunctional properties that then are important for processing, or even if I want to use these materials at home, how do the dispersion conditions change the properties? Does it matter whether I put my powder into water for an hour or 24 or something? So anyway, so first of all, I would like to introduce her work where she changed the extraction process. So we used the classical wet extraction process. So we started with a stone-milled quinoa flour that we procured in the UK. And it's got like a composition here that we determined. So we took the quinoa flour, and the classical approach is you put it into water at high pH to solubilize the protein component. And then what normally happens next then is you would do a hexane extraction to get rid of the lipid component. So here we can see there's 6.8% lipid in this quinoa flour here. We decided we don't want hexane, so we centrifuged the material and removed the cream layer because the oil bodies would cream, and we removed those. And then we did the next traditional step, isoelectric precipitation of the supernatant. So at pH 4.5, the proteins all come out of solution. And we did this with 1 molar hydrochloric acid, which is what is normally used and reported in literature. And we also used more cosmotropic acids. And the idea was maybe preserving native protein. We had we wanted to preserve native protein for our structuring approaches. So then you centrifuge, you rinse and precipitate, and do this, neutralize and freeze dry, and have your quinoa protein isolate powder. So we have these 3 materials now, and I apologize for showing a table here. So we're having these 3 quinoa protein isolates, and the protein concentration is, it wasn't 80%. We had much less than that, or slightly higher for using these acids, but not massively higher really. The big difference was in the lipid component. Using the more harsh acid, we recovered in quotation mark because we weren't really interested in the lipid, but we found less lipid in our isolate than by using the other two precipitation acids. So if you're interested in carrying over lipid for nutritional reasons, there is really a point to be made here. Here we reckon the hydrochloric acid had an effect on the oil bodies where the lipid is contained in the flour, but these would have been already damaged in the stone milling process. So we shouldn't forget that actually the primary process also might have a huge effect on the outcome. The protein yield was pretty much the same. So we have now compositionally different quinoa protein isolates. And we looked at soluble protein, measuring using a certain protocol taken from literature. I was just at EFOS last year. There is now a cost action in Europe where people are actually wanting to look at, because everybody measures is slightly different. How are data ever comparable? How is this ever going to be useful, for example, for industry? So I think, well, I wish them good luck because it's a huge challenge. I signed up for it because it is a, you know, and when what can you actually standardize between different labs, etc. So that's quite interesting. But anyway, soluble protein measured after 1 hour of gentle agitation at 22 degrees C for 1% solution or dispersion. We can see we have differences between, depending on the precipitation acid we used. So we have much more higher soluble protein when we use acetic acid and citric acid. Later I will show some data on structuring processes where we probably still hypothesize that soluble protein is important, but there's still a lot of open questions. And equally then, if we take a 1%, if we measure solubility at a certain pH and increase protein concentration, the solubility goes down if you're using this solubilization process, but it's still higher for using these weaker acids. Okay. So here we have, so this is the data basically I just showed solubilized for 1 hour. These differences disappear if you gently stir the 1% solution for 24 hours. So that's just interesting to note. I'm not so sure I understand that. And then, of course, something else will happen if you have salt present in your solution or in your solvent. On the right-hand side here, we see some thermograms. So we're heating these samples up then in a differential scanning calorimeter, and we can see the area, well, above the curve here, really. We can see this is lowest here, and the area gives us an idea about how much native protein is there. And here it's higher for these samples here, and we have a higher amount of native protein. So that worked, and that also explains obviously why the solubility is higher. So moving on, we are interested, we then looked at the gelation behavior of a 10% preparation after we dissolve just for 1 hour. And this is a classical test then you would do. You stick it in a rheometer, you oscillate the sample, and you measure the viscous and elastic response. And if you focus just on the solid symbols here, which is the elastic response, so these dispersions at low temperature have basically no structure, but once the temperature is high enough that the protein starts denaturing, we get some structure, or the still native protein denatures, we should put it that way really. We're getting gel formation. And classical for plant proteins is that the gel formation or the texture creation continues upon cooling the sample. Oops, sorry. So here sorry, confusing here. So we're holding the temperature at 95 degrees C, we're cooling down, but structure continues to evolve. And sort of as expected for these two materials here, we get a higher final elasticity value, or gel strength, if you want to just call it gel strength. So, as I mentioned at the beginning, this gel strength is of course related because we're having 10% preparations, all three of them. So the differences in gel strength will be related to difference in microstructure. So the data I just showed relate all to this column here. And these are confocal laser scanning micrographs. The green stuff is the protein, and red is the lipids. So the lipid that we carried over. I can't remember how big the scale bar is, probably about 20 micron or something. I can't see that. Yep. So these red spots here they're larger than what an oil body would be in the plant source. So basically the milling process, the original milling process, and then maybe also the extraction process has led to coalescence of the natural oil bodies. So we had some reformation of structure going on, but we can see different network densities here, and this is the gel strength, the final value for the gel strength I just showed on the earlier slide. So if we then change the solution conditions, we can see different gel structures or different microstructures and also different final gel strengths. If you have salt present, we get, for example, for this material here, it's basically not a gel anymore. So we have a very porous system that's probably more like a dispersion. So there's a lot happening, and all of this will feel very different, will be relevant to different product applications. So we then thought, okay, can we sort of recover the lipid bodies, or can we get rid of them, or at least not destroy the protein that's stabilizing the lipid bodies? So we did some work. We cryomilled, or the student did. She cryomilled some seeds. So these are quinoa seeds that were obviously not the same the stone milled flour was made from. Whatever happened here. Okay, so we cryomilled seeds, we did the same extraction process, and what we now have, we have a much higher protein yield. So through cryomilling, we can get much more value in our material. We're having now nearly 80% of protein, or maybe nearly 75% of protein, and we still have lipid carryover because we still didn't use hexane extraction. However, no, there was no difference between the 3 different extraction processes. So we can see a huge impact of the primary process. I'm going to the next slide. These are now the microstructures of these samples that were also 10% in weight. And 1 hour gentle as this sort of dissolution regime here. And we can see on the left-hand side, we have a very, very different microstructure to what we saw beforehand. Much more open, much more porous, even though actually the final gel strength value is not too different. But interestingly, for the other 2 extraction acids, we have now much denser networks, and we're having a much higher gel strength. So the primary process has not just an impact on the composition, but then the composition has an impact on the final gel strength, and probably also the properties, the more natural properties or native properties of the protein. This is all I really wanted to say about the quinoa protein isolate work. We started doubling about 2 years ago with free structuring, directional free structuring. This is a technology that is used to create porous, unaligned and anisotropic materials. And these are then often, for example, made of agar or collagen, but they're also used in sort of other mechanical material type stuff. These free structured materials are then often dehydrated to kinetically trap these anisotropic microstructure and then used as scaffolds, for example, in the healthcare sector, or actually to grow cultured meat. Literature is sort of emerging to create meat analog textures using this technology. Formulations invariably contain a polysaccharide additive to actually, when you then melt the structures, because if you want to make a moist meat analog, don't dry it, that we have a self-supporting material. So we need to add something in. There is a lot of open scientific questions, what is going on? And we set ourselves a challenge, or that particular PhD student decided, I don't want to add any polysaccharide additive to my materials. So we set up a process we're using, and this is obviously in the moment very much a batch process. One of the big criticisms is that free structuring is not scalable,
but who knows what the future brings. So we have an insulated mold, and this is 30 millimeter inner diameter and about 50 millimeter high. We put this mold onto a cold shelf, our protein dispersion, so these bubbles here are our globular protein, and the ice crystals go directionally because this is an insulated mold. The challenge is that the temperature gradient in this direction will decrease, so ideally we should control this temperature over time so that doesn't happen, but this is published in literature. We haven't put that in place yet. We work with 3 different commercially obtained protein isolates. And in terms of kinetically trapping the structure, we have, oops, this is just odd here, isn't it? We have freeze-dried our material as well, and this would be mainly for the purpose of imaging with micro-CT. We can't image wet samples in there because the contrast between the polymers and the water isn't high enough, so they can't see the structure then. We have also pre-treated the pea protein isolate dispersions with shear and temperature. And if you then use high enough concentrations, we get a self-supporting material. Alternatively, we have melted these material structured samples in calcium chloride solution to sort of enforce the bonds between the protein, and we're getting a self-supporting material at the end. So the functional properties that are discussed in literature, and we can confirm that influence the free structuring process are, well, here we have open questions here, to be honest, but soluble protein will depend on the pH, so the more alkaline, the more soluble a commercial pea protein isolate will be. Water holding capacity, so how much water can it take up, might also be important. And for samples that contain 20% solids, so we're at 80% water, and we looked at 3 different pH values and determined freezable water, so how much water in the sample is available to freeze in our process. And freezable water is important. So on the left-hand side, we have, this is now a freeze-dried sample made of 20% solids, and we have a nice, we have an anisotropic texture here. So we created our material, freeze-dried it, and then broken this open. We can clearly see an anisotropic texture here. I do admit it still doesn't look like a meat yet, but it's a start. So if you had less than 50% freezable water, we don't see anything like it. So in a moment, 50%, we found this for other protein isolates as well. It's a little bit of a magic number. It's, of course, only 2 minutes. Okay. Started late. Anyway. Okay, so I need to leave out some things then. Okay, so anyway, it's a magic number in the moment that we need to understand. Okay, so if you now melt these structures in calcium chloride, because if you just melt the structures, then we get like a protein dispersion back. But if you melt these in calcium chloride, we're seeing these sort of textures here. And if we break them open, we can see increasing fiber formation here, or thicker fibers. And in this direction, also the solubility of the protein increased. Okay. So here's an image of the microstructure of one of these samples. So at the bottom here, we can't really see much by eye. At the top, we can see these sort of sheet-like structures here. And here we cut through the sample. You're looking like that way through the sample. So this is at the top surface of a cube cut out of this middle here. We can see we're having these sort of non-spherical structures. The white is the protein. And this is due to us not controlling the temperature gradient. But at the bottom, we can see these sort of spherical structures. So the ice crystals grow, they're nucleated, they grow needle-like, and then they grow bigger. So I then finish soon, Anna. So we look then at the compression strengths, because we're interested in, we can analyze the images, of course, and we're still working on that. But we looked at the texture, so there is measuring textural anisotropy, so compressing the samples in the direction of the fibers and then perpendicular to the fibers here. And we can see the textual anisotropy. Well, these data here confirm the anisotropy as well. And out of fun, because what you then have to be also turned the sample round on its head because of the different textures at the top and bottom, and we can still see the same result. Okay. So we also did a heat and shear pre-treatment with 30% weight percent solid samples. These had 50% freezable water, and I'm only showing the freeze-dried samples here. So this is a different pea protein isolate. Its natural pH was 6.8, and here's the solubility. And we also looked at the same pea protein isolate at pH 9, had a higher solubility there. And what we can see actually at this lower solubility here, with a higher textural anisotropy compared to the sample here. But overall, the textures were much stronger. So is solubility important? Is solubility not important, or soluble protein? Here we have, this is a different commercial protein isolate that's very, very soluble, but we can't even grow any anisotropic textures here. Here we mix those two protein isolates. We have a similar solubility, and we're getting then similar texture responses here. I think given that you're urging me to finish, shall I stop here? Before I... maybe just a quick comment to extrusion processing. So Kati has talked about this. This is the textures here she presented. There is this idea of molecular alignment in the extruder, molecular alignment creating these fibers. There is also very new data in literature that proposing actually we have the globular proteins. It's a chain-like alignment, etc. So maybe just to sum up, I don't think it's if you work with, it's not understood how these structures are actually created in the extruder. There is different schools of thought, different evidence for different things, and it's really super challenging. And you don't necessarily need high temperatures either. So we have extruded our mixed protein system at 90 as a maximum barrel temperature of 90 degrees C, and we had a lovely fibrous texture as well. So lots of challenges. You can do a lot of trial and error, but that's not the future, is it? Okay, I'll stop here then. Okay, thank you.
Thank you very much, Bettina, and I'm sorry to rush you. To have rushed you.
I think you are rushing too fast.
Yeah. Do we have some questions?
It's a bit too different.
Thank you. I'm not sure if I understood everything, but
no, sorry but
may I ask? I'm just thinking the protein isolates. Many times this sodium chloride is used, and then there's sodium in the end product as well, which, from nutritional point of view, is sort of a problem. So, do you think with different processes you wouldn't need to use sodium?
I'm unsure what the sodium chloride is used for. Sorry.
Okay, I think you had it. Did I got it right? You had in how the protein concentrate was, or the protein isolate was prepared.
That was precipitation acid,
yes.
Hydrochloric acid,
yes.
and there wasn't any sodium chloride present there.
Okay.
So sorry about that now.
But there was in one dispersion.
Yeah, in the dispersion thing, yeah. Well, I don't think, it's an illustration. I mean, lots of food products have sodium in there, so it's always good to do an experiment with a small concentration of salt present, but it wasn't actually that beneficial.
Okay.
Thank you, very nice presentation. So the melting in the calcium chloride step, how did you arrive to the calcium chloride? Was that or did you try other things as well that works?
I don't think we tried other things. We did, calcium, what's the right word? I don't know what the right word is right now in my head. Sorry, it's sort of, catalyzes is the wrong word, sorry. It helps with the double disulfide and hydrogen bond formation between the protein. Is it molecules? Is it aggregates? So we're still looking into that. But and also, of course, then the question is, okay, now you've got a lot of calcium in the product. Is that any good? But that's the next question. No, we haven't tried anything else. That actually, we used sodium alginate, and then we wanted to move away from having this polysaccharide additive, which is then also on the product label, etc. But yeah, but where exactly it works, but I do believe we've quantified which bonds were formed as well, or yeah.
So what would you recommend?
What would I recommend?
For a future process?
For a future process? Well, I mean, I guess in the moment extrusion processing is the, but yeah. I mean, free structuring, I know other groups are looking into free structuring. The big challenge is we don't really understand the material properties or the system properties. You know, it's not just the protein ingredient, it's how does it behave in the environment, etc. What is really important. But I think we are, well, I know there's other groups looking at it as well, and we have some ideas of making it, we're hoping ultimately to make it a continuous process as well, but I think we're a long way off that yet. But of course, free structuring will not have the sort of detrimental effect on nutrition or effects on nutritional properties. So, but of course, it will also not deactivate anti-nutritional factors. So there's a lot to consider. So I think we are still playing. So anyway,
very good.
Thank you.
We'll give Kati and Bettina a big applause.
Yeah, by that we end the symposium. We started with an overview how important plant-based proteins are as a part of our transforming the food system. The vision of PEN Sweden is that sustainable produced food with health benefits is the consumer's first choice. And we have seen that it is not that simple yet. I mean, we have to prove the health benefits. We have to prove, of course, also the sustainability. But to be the preferred choice of the consumer, it should be taste at least as good as the alternatives and not be too expensive either. And that also for the entire world. So we see that we still have some challenges, and researchers like that, because otherwise we have no work to do. So I think we had some good news. We had a little bit of challenges also to see, but it is good that we know these challenges and can work on that as good as we can. By that, I would like to thank the whole faculty of organizing this, and also those behind the scenes as Johnny Carlson, Annette Oskarsson, Nina Jansson and Lucas, who also have worked on this symposium and to be successful as it was today. By that, I wish you safe travels back home, and thank you.